Good morning, everyone, and thank you for coming. And thanks a lot, Adria and Yuris, for organizing this really interesting session and really bringing us together on what has not been a unified concept, but is now. The idea, and also for the opportunity to present here, the idea of socially transmitted fluids is really cool. These molecules, although very different in specific functions, share some very big picture parallels that are important in considering their evolution. These fluids can range from things like milk in mammals through venoms in certain species. And as Adria said, they're molecules made in one animal that exert their effects on another. But how do they do this mechanistically, and how does this evolve? So from the standpoint of the delivering animal, making and transferring these molecules must be evolutionarily advantageous, even if it does come at some kind of cost to the producer, whether it's energetic in many cases, or even loss of your head or your life if you're a praying mantis male. But the situation is, I think, more nuanced when you consider the recipient. And the effects can range from apparently advantageous, like the recipient of milk, for example, to apparently very disadvantageous, like the recipient of a venom. And seminal fluids, which is what I'll be talking about, lie, in my mind, somewhere in the center of this continuous to very advantageous to very disadvantageous. These are molecules made by males, transferred to females during mating, and they mediate changes in the female and interactions between the sexes, which can include sexual conflict, competition, but also cooperation. So they can have both positive and negative effects on the female recipient, all of which shapes their evolution. So in my talk today, I'm going to start by talking about these apparently positive and apparently negative interactions, which, as you'll see, are less positive and less negative sometimes than they look. That's my first theme. And then my second theme, which is that to really understand their evolution, you have to understand their mechanism and the whole pathways in which they act. And I'll talk a little about that as well. I'm from the US right now, and there we are extremely focused on presidential whatever. So I named my talk Team of Rivals, which I am trying to advance. Hold on, I think I can get it, yeah. After a book by Doris Kearns Goodwin on the Lincoln administration, which she claimed was so successful because they included people who agreed with Lincoln and people who were opposed so that they could bring both points of views in. And I thought that was a pretty good explanation of at least seminal proteins and maybe other molecules as well. So I'm going to focus my talk on the role of seminal proteins, primarily in Drosophila melanogaster. And in particular, I'm going to focus on one particular seminal protein, a 36 amino acid peptide called the sex peptide. The sex peptide exerts many effects on the female. In this slide, modified from a beautiful review by Hopkins and Perry, you can see that it can you see my pointer? No, you'll just have to read it. There's no pointer here, so just take my word for it. On there, it says that among its effects are increasing egg production by the female, changing her feeding behavior, changing her gut size and physiology, decreasing her receptivity to future mating, increasing her aggressiveness, and many other effects. So if you think about these various effects together on the female, it's quite nuanced. Some of the effects are, pretty clearly seem positive to the female. For example, coupling the increase in egg production to her receipt of sex peptide seems like a very positive thing to do. On the one hand, it improves progeny production by both sexes. On the other hand, it allows the female to couple her increase in this energetically demanding process to the receipt of sperm and seminal fluids, which is the only time that those eggs will actually be able to develop when she has sperm. Aggressiveness over food, reported by the Whigby lab, is another thing that's advantageous to the female, presumably because she can get more food if she's more aggressive. It's actually interesting because it tailors the social environment since she's aggressive against other females. So this is saying that this socially transmitted molecule affects not only the recipient, but also the whole environment. There are other things that sex peptide does, controlling the rate of release from storage, controlling gut growth, where you can tell similar stories that it would be advantageous to the female as well as the male. But there are other effects of sex peptide that appear to be disadvantageous to the female. Effects on immunity, on longevity, or maybe the most famous effect, which is that it makes females less interested in mating and less attractive. This initially seemed like a very counterintuitive thing for the female, although it's great for the male, because as part of sperm competition, she doesn't mate with anyone else or so on. But a recent paper from Kohlmeier et al. From the Billiter lab shows that it actually may be advantageous to the female, despite my negative arrow. They showed that really what happens is the female becomes choosier after the first mating. So she just gets sperm so she can have sperm, but then she becomes choosier, and if a better male comes along, she'll mate with him and his sperm will take precedence. So it's actually sort of negative, sort of positive. This process also affects the social environment in that the lack of interest of females in males affects those males, and the male's ejaculate itself also negatively affects other males in that if they do succeed in mating, molecules in the ejaculate and the sperm can affect the likelihood of the second male siring progeny. So I presented this to show that there's a sort of complex web of cooperation, conflict, competition, with effects on the female and on the social environment. This is pretty complicated, but there's more. And I'm going to tell you one more story and show two more arrows that will seem counterintuitive. So right now it looks like this is all pretty good for the male and not too bad for the female either. But it's all, I've presented it sort of in a male manipulating the female idea. But there's several modifications to this. First of all, work by Snigdha Misra in my lab showed that sex peptide, the seminal protein from one male, which supposedly disadvantages the next male, can actually bind to the sperm of a second male. And so here you can see sperm stained for DNA, and here you can see the sperm from a second male binding the sex peptide from the first male. And that is not just a picture, but it's actually real biological effect. In red is the one on the left is a wild-type male's mating propensity, progeny propensity. In the middle is the number of progeny that someone without his own sperm, without his own sex peptide. I'm sorry, I can't claim jet lag, but I'm still tired. And here on the bottom are the same kinds of males, but the blue is when that second male received sex peptide from a first male. So it can actually improve the fertility of the second male. So we're adding some arrows now. What I just showed you is that a male not only can disadvantage another male, but he can also help another male. And then I don't have time to show you this, but our biochemical work shows that the female can actually do some biochemical things that help the action of the sex peptide within her. That's both shown by Finlay et al. Using evolutionary rate covariation and by some physiological work that Snigdha Misra did in our lab. So this is the web so far. And what I want you to see right here is that it's not so clear. It's not always positive. It's not always negative. And that's where I want to stop this part of the talk and switch to the second part, which is, okay, great, how does this evolve? So given the important functions of seminal proteins, some of which are in fact advantageous to males and possibly not so good for females, one might expect that their primary sequences might evolve rapidly. And that is indeed true for about 15% of seminal protein genes. Seminal protein genes are the black dots, and the round ones are not reproductive gene controls. This is work by Willie Swanson in our lab some time ago, and he showed that about 15% of seminal protein genes evolve rapidly, One of these, for example, is a molecule called ovulin, which we know is important for ovulation, and yet it evolves very, very rapidly. This is true, but in addition, there are other loci or other targets of selection, which is not just that the primary sequence can evolve rapidly, but seminal protein genes can be gained or lost over evolutionary time. This is just showing loss over here. So that's great, but Civetta and Putlar, looking at the whole suite of about 300 seminal proteins, found that although what I've told you is correct, it only applies to some seminal proteins. Others seem to be evolving by relaxed selection or under constraint. So what about their pathways and their effects? So for example, Hopkins et al. Found in a paper published this year, found that sex peptide, who I've just been telling you a lot about, its active region in the processes I've talked about is widely conserved across Drosophila, which was really quite a surprise, although its copy number varies. And so what else could be a target of selection then? So what I would like to argue is that the pathway through which these molecules act may be other targets of selection. I'm going to give you two examples. First of all, the receptors can be targets. So on this slide, on the left, is work from Yapichi and colleagues in the Dixon lab who discovered the G protein coupled receptor for sex peptide, which is called SPR. And what you can see in the blue are all the controls, and in the red and yellow in the middle are cases in which they knock down the sex peptide receptor. And you can see when it's knocked down, females do not increase their egg production after mating the way they normally do, which is one of the things that sex peptide induces. So this receptor that means 2 more minutes. One more, I'll have to talk fast. Okay, this means that sex peptide receptor is obviously important in sex peptide action, and that's actually been shown in 2 other Diptera, 2 species of olive fly in the genus Bactrocera, and in 2 Lepidoptera, which is much farther away, such as the cotton bollworm here. You can see from Hanin et al. That in wild type there's a lot of eggs, and in the knockdown there's almost none, and Spodoptera, another lepidopteran. So how about other Diptera? So recently my colleagues and I in mosquito, so Laura Harrington and her lab and Connor McMenamin and his, knocked out sex peptide receptor from the Zika dengue mosquito, and it doesn't have any effect on egg laying post-mating. So we have some Diptera where it does, some Diptera where it doesn't, some non-Diptera where it does. So clearly there's evolution going on here. What is actually going on, we don't know, but there's work from Toshiro Aigaki that in Drosophila that suggests one possible correlational explanation, which is that species in which sex peptide receptor has acquired expression in the reproductive tract, those respond to it for post-mating responses, and those where it hasn't don't. So the second place that we think could be an evolutionary target is in a different network in which sex peptide acts. So I'm going to skip over this slide and just say, look at the graph and see in the red that the gut can grow, and it grows for a long time, almost a week, which is almost a whole generation in fly. This long effect of sex peptide is due to the fact that the molecule binds to sperm, which retains it in the female for up to 2 weeks. I'll do this fast. We have identified a part of the pathway that does so. We've been working on the biochemistry of the pathway. You can see here on top sex peptide being presented to the sperm to bind it, which it will do with the rest of the pathway. And really quickly, just like Hopkins found in the column here, that there's variation in the number of copies of genes for sex peptide, so Jolie Carlisle and Ben McCormick in the lab have found variation in the number of copies or presence of those pathway genes in different species. So we're currently examining the binding of sex peptide to sperm across Drosophila taxa to see whether and how the presence or absence of members of that pathway relates to the binding of sperm. So in summary, conflict, cooperation, and competition act holistically across an entire pathway, not just on the socially transferred molecule. Some parts of that pathway may be more evolutionarily labile than others, and so to understand the evolution of the pathway and of the socially transferred molecules, we really need to understand the mechanisms and interactions that are involved. And briefly, these are the people whose work I told you about. They're all wonderful, and I will not go into detail, but they all did beautiful things that I hope I've been able to explain. So with that, I'm happy to take questions. Sorry to run to the gong.
Thank you very much. While I set up the next, you can take maybe a question.
Sorry about that. No problem. Yeah.
Thank you for that really lovely talk, really interesting stuff. Just very briefly, do you think that other alternative targets might be the regulation of some of the metacinus pathways? Things that are hard to find, right? If you don't find variation in the primary sequence, it might all be to do with how it's regulated.
Yes, I think that's absolutely clear. The question is, could the evolutionary target be the regulation of members of the pathway as opposed to the sequence or amount? Absolutely, and I think that Igaki's data saying that when the receptor's in the tract, there's an effect, and when it's not, there isn't, is the first sort of example of this that I can think of. The other ones that we work on are all in the reproductive tract.
God.
We have time for another question. We can make this look deliberate.
Yeah. I am struggling.
Yeah. That binds sex peptide to sperm. By genetics. So we did knockdowns or knockouts of various seminal proteins and tested whether the effects of sex peptide persisted. And in cases where they didn't, we then tested whether it affected the binding of sex peptide to sperm. I'm sure that the pathway is not complete yet. There are 300 seminal proteins. We've tested 50 of them, so I'm sure there are going to be more, including some that have themselves bind to sperm, which are our tank.
Yuqi, I would like to share with you my study in larval storage proteins and metabolic division of labor in ants. Metabolic division of labor. Okay, if we think about how life evolved from simple to complex to amazing different forms we see today, it relies on cooperations between different units within an entity, where the increasing division of labor plays an important role. It can be observed across levels of biological organizations, such as organelles within a cell, between cells within a multicellular organism, between microbes in a community, or between multicellular organisms within a social group. Today, I would like to focus on the metabolic division of labor in ants. The metabolic division of labor is defined as the partitioning of a given metabolic process into several elementary metabolic tasks performed by separate units within a cooperative entity. We believe that ants may provide a unique insight into the evolution of metabolic differentiation, not across cells, tissue, or organs, but between different individuals making up a superorganism. So, as a superorganism, the colony behaves as a reproductive unit. Therefore, each individuals in a colony are like cells in a multicellular organism. And from an experimental perspective, ants are amazing. The individual ants are accessible, and their socially transferred materials are also accessible. As a species-rich taxa, there are different ant species display trophallaxis, also known as social regurgitation, to a very extent. So here is a video showing trophallaxis behavior in ants, where you could see an ant who is full of fluorescent in her crop passing this lit-up content to another individual, which is pretty amazing. So, we usually term such social regurgitation as trophallaxis. It actually includes many different social transfer and broadly defined as the direct ingestion by one individual of material excreted, secreted, or regurgitated by another. Fluid passed by social regurgitation contains proteins, RNAs, hormones, and more. It builds a social circulatory system connecting every individual in the colony, and it also affects larval development, collective decisions, and more. Therefore, we think that trophallaxis enables metabolic division of labor in ants. So, if we go one step further, what content in trophallactic fluid could underlie metabolic division of labor? Well, we think larval storage proteins, LSPs, are one of the strong candidates. The LSPs are produced by the adults and secreted into trophallactic fluid in some ant species that rely on the trophallaxis network for resource distribution. And what makes the LSPs intriguing is that the LSPs are well known and rich in larval hemolymph across all insects, and it is an important protein for storage before metamorphosis. Social Hymenoptera and bees wasps evolved from parasitoid wasps that lay eggs in the bodies of other insects, suggesting that they rely on larval hemolymph components ancestrally sourced from the prey. These feature altogether suggesting that LSPs are consolidated resources to pass around and make LSPs a good candidate to mediate metabolic division of labor. So, our research question is, are LSPs mediating metabolic division of labor in ant colonies? To test our questions, we chose two species which are representatives of two clades of ants, Ponerine and Formicoid. For the Ponerine ants, we chose Platytheria mocharici, which is a species with traits and behaviors that we believe the common ancestral ants have. In mocharici, the adults bring the prey to the larvae, where the larvae will eat the prey directly, and the larvae, very interestingly, they have specialized tubercles which will secrete fluid for the adults to drink. For the formicoid ants, we chose Mimica rubra, which the nurse and foragers can be distinguished in this species, and it also has a bigger colony size. Most importantly, it shows a typical trophallatic fluid network where the adults, mostly the nurses, feed the larvae with trophallatic fluid. Although the larvae were found to regurgitate and excrete to adults, the biological meaning of this behavior is not clear yet. Well, overall, the reason we chose these two species is that they show a reverse nutritional flow, where in the mocharici, the larvae can be considered as a digestive caste of the whole colony, while in rubra, the nurse is the digestive caste. So here is a picture and also a short video showing where the tubercle of the larvae from mocharici is, and also the tubercle feeding behavior. As you can see here, the adults will gently massage the larvae to make the larvae give their tubercle fluid so that they can feed on them. So, how do we test if LSPs are mediating metabolic division of labor? We want to know where are they produced by using transcriptomics. We also want to know where they end up by using proteomics. So, more specifically, for two species, we would sample the tissue from adult and larvae of mocharici and nurse forager larvae from the rubra. These tissue samples will be used for RNA-seq and also for the proteomics. Also, we would sample the fluid of interest from all these different individuals, like the crop content from the mocharici adult, the tubercle fluid from the mocharici larvae, and the trophallaxis from nurse and foragers of rubra, also the regurgitation and excretion of larvae from Mimica rubra. So if we plot the LSP abundance against LSP gene expression to test our hypothesis, which is that whether LSPs are mediating metabolic division of labor, we would expect to see the Mycorrhizae larvae have high LSP expression and high LSP abundance in their tissue, as well as in their tubercle fluid, while the adults of this species don't produce any LSPs at all, but one could still find LSP in their crop content. Like the gene expression level will be 0, but you will still find the proteins. Well, for rubra, as it has a opposite nutrition flow, we would expect the larvae still produce and have LSPs in their tissue, but would not give much in their fluid. While the nurses will produce a lot of LSPs and secrete them into the fluid, but not reserve for themselves in the tissue. So we would expect something like that. And for the foragers, we actually don't expect them to express any LSP at all, or having an LSP, maybe some in their tissue. So what are the genes we are looking at precisely? This is a gene tree of LSPs in 24 species of ants plus Drosophila. There are generally 3 LSPs in ants, which we are calling Hematognoptera LSP 1, 2, and 3. They are all related to more to LSP2 in Drosophila than to LSP1 alpha, beta, and gamma in Drosophila, as you can see here and here. And I also highlighted the genes in rubra and Mycorrhizae in this tree, so you can see their distribution. So what do we found? This is a correlation plot you're looking at with gene expression on the x-axis and fluid protein abundance on the y-axis. And different casts are color-coded. The clouds you're looking at are all the genes that we found in the PMOC fluid. We have pinpointed and also faceted the 3 different LSPs. Each black dot here is a biological replication. So what we found is that, as predicted, the P. Mycorrhizae larvae are producing a lot of LSPs and secreting them into their tubercle fluid, while the adults are drinking this and receiving LSP1 and LSP3, especially from the larvae. So if you look at these 2 bottom graph here, you will see that the adults are not expressing any LSP genes, at least high LSP1 and high LSP3, but they still have these LSPs in their crop content. While surprisingly and interestingly as well, some Mycorrhizae adults are producing high LSP2 and secreting them into their corp. Well, the next graph is the similar type of plot, but now on the y-axis is the protein abundance in the tissues of larvae and adults, now the fluid. So what we can see here is that the larvae, as expected, are hoarding some of these high LSPs in their tissue, while the adults are producing some high LSP1 and 2, especially high LSP2, and reserve them into the tissue, but they are not producing high LSP3 at all. But interestingly, they still have this high LSP3 in their tissue, which could either be from the tubercle feeding that they get from the larvae tubercle fluid, or the leftover from their own larval and pupae phase. So in Mimica rubra, we expect a different direction of nutritional flow. So indeed, we see that the nurses and some foragers actually are producing LSPs, especially high LSP2, and secreting them into the fluid. While in larvae fluid, we noticed that some individuals don't put any LSPs into their fluid, which met our expectations, while some do so, especially in their excretion, which we don't know the reason yet. Lastly, the idea of nurses and maybe also foragers are producing LSPs and secreting them into the fluid is supported by the high expression but low tissue LSP abundance level in the Mimica rubra, especially LSP2. As you can see here, the adults are producing a lot of the LSP2, high LSP2, but they are not reserved them into their larvae, into their tissue. In other words, that means that they are giving them away with the fluid to the larvae. So in conclusion, we found that P. macrorhizae larvae are secreting LSPs into their tubercle fluid, and adults are receiving these proteins. And surprisingly, some P. macrorhizae adults still producing LSP2 and secrete them into their crops, as well as reserve into their tissues. And for Mimica rubra, the nurses are secreting LSPs, especially high LSP. We believe that. So in conclusion, we found that P. macrorhizae larvae are secreting LSPs into their tubercle fluid, and adults. With that, I would like to thank all my collaborators and the.
There we go.
Hello everyone, my name is Carneiro. I'm a second year as a PhD student at the University of New Brunswick in Canada. And under the supervision of our Nuduku, we have this project in which we want to address how cancer cells can cooperate by secreting materials and respond to it to increase the tumor progression. So the evolution and stability of a multicellular system is dependent on the establishment of cooperative interaction among cells, and the loss of this cooperative interaction between somatic cells can result in an uncontrolled proliferation and ultimately cancer. So in this context, cancer cells are seen as cheaters that has expressed selfish behavior relative to normal cells, but they can cooperate with each other to enhance, like improving their fitness and ultimately contributing for tumor progression. For instance, in a heterogeneous tumor, cancer cells can cooperate by secreting molecules that neighboring cells will respond to. And indeed, some studies have linked such cooperative interaction with tumor progression by tumor growth, drug resistance, and metastasis. So metastasis, as you might know, is the last stage of the tumor progression and refers to the spread of cancer cells to other parts of the body. But to reach the vasculature and disseminate, cancer cell has to acquire the ability to move and invade new organs by breaking tissue basement membrane in a process that is called epithelial-mesenchymal transition. And many factors are known to induce this transition, but a key protein secreted by both normal and cancer cells is the so-called transforming growth factor beta, or TGF-beta. The classical view of metastasis envision single cells leaving the primary tumor, migrating and invading neighboring tissues and disseminating via bloodstream to distant locations. However, more recent findings suggest that some of these steps might in fact involve a heterogeneous groups of cells that with distinct characteristic that cooperate for successful dispersion, colonization, and then poor prognosis. So with that, our project will address the possibility that cancer cells can cooperate by secreting and responding to molecules to increase their metastatic features in terms of migration and invasion potential, investigate the mechanism involving such interclonal cooperation, and address the generality of such cooperative interactions. And to reach these goals, I used several cancer cell, lung and breast cancer cell lines with different metastatic potential. And because as in other systems, this sharing, exchanging, and transfer of materials involve materials, I collect the media in which the most aggressive cell line has grown and secrete materials, secrete molecules, the so-called conditioning media, and expose the less aggressive cell lines to this medium and examine the invasion and migration potential of these cells. Also to investigate if this interaction has a synergistic effect, if it's like both way cooperation. I co-culture these two cell lines together and evaluate the ability of these cells to invade. So first I will present my data on the breast cancer cell model that includes two different cell lines that represent the most common type of breast cancer. And they differ in the metastatic potential. So on the right, you see MDA MB231, or just MDA from now on, is known to be more aggressive than the other one. They produce, secrete, and respond to TGF-beta. Again, the key molecule that is known to induce EMT, or epithelial mesenchymal transition. And on the right, MCF7, that is less aggressive. They respond to TGF-beta, but they don't produce that. So then to confirm that MDA can secrete and respond to TGF-beta, we used a TGF-beta receptor inhibitor. And as expected, the migration of MDA was decreased in contact with this inhibitor. Then to address whether MCF7 responds to TGF-beta secreted by MDA, we treated MCF7 with the conditional media from MDA and compare its response to exogenous TGF-beta as a positive control. And in both cases, both exogenous TGF-beta and the conditional media induce phenotypical changes that are associated with the epithelial mesenchymal transition that leads to an increased migration. Regarding mechanism, consistent with the hypothesis that MCF7 can respond to TGF-beta secreted by MDA as a soluble factor in the conditional media, we found that by blocking the TGF-beta receptor on MCF7 decreased the effect of MDA in terms of migration. Similarly, when we talk about invasion, MDA conditional media also increase the ability of MCF7 to invade. And then to evaluate the possibility that this cooperation can be synergistic, we co-cultured MDA and MCF7 together and evaluate the ability to invade. And both cells had increased invasion when they were in contact. This suggests that by promoting the invasion of MCF7, MDA cells also enhance their own invasive potential. So in addition, we found that the invasion of both clones in co-culture required the ability of MCF7 to respond to the TGF-beta secreted by MDA. Since by blocking the TGF-beta receptor on MDA cells alone leads to decrease the invasive potential of both cells. That is, MCF7 depends on the TGF-beta secreted by MDA, but MDA increased invasion is also dependent on the ability of MCF7 to respond to TGF-beta. So based on these our findings, we propose a model in which a highly metastatic clone can co-opt through TGF-beta crosstalk a weakly or non-metastatic clone into expressing a pro-metastatic feature that will in turn enhance the invasive potential of the former, through which we call help me help you strategy, and ultimately result in an increase in the overall invasive potential of the tumor. So next, we want to address whether our findings with the breast model can be extended to other type of tumors. So we developed this second model system that we used two lung cancer cell lines that derive from the same metastatic line that grow as a mixture of cluster cells in suspension and adherent cells. And since we knew from previous experiments that AS, that we call the adherent suspension, it's more aggressive because they do produce TGF-beta, while compared to SS, the suspension selection, that it's less aggressive, so they don't produce a TGF-beta, but they do respond to it. We co-culture these two cell lines together and evaluate the ability to invade. So we found that the total number of cells invading in co-culture were higher when we compared to the sum of each cell type grow as in monoculture, suggesting that there is some kind of cell interaction that increase the overall invasion of the tumor. So lastly, to address whether these interactions are possible even among different clones, different tumors, we treated the breast and the lung line with the conditional media from the opposite cancer type and assessed their ability to migrate. And interestingly, we found that the conditional media from the highly aggressive lung line induced migration in the less aggressive breast line, and consistent with the possibility that this effect is involving TGF-beta, the migration of the breast line was decreased in the presence of the TGF-beta receptor inhibitor. Then, to test whether the conditional medium from the highly metastatic, highly aggressive breast line would affect the highly aggressive lung line, we also used the conditional media and found that a significant increase of the migration of the lung line when in contact with conditional media from the breast cancer line. And again, the effect was decreased when they are in contact with the TGF-beta receptor inhibitor. So overall, we argue that the synergistic cooperative interaction can easily evolve between a highly metastatic clone that are able to constitutively secrete molecules that induce and maintain their own metastatic features, and a nearby clone that express cognitate receptor to those ligand and can switch to a malignant state phenotype. So such cooperative interactions via transfer materials provide a competitive advantage for both clones and result in an increase in the frequency and overall change the metastatic potential of the tumor. And finally, we suggest that this interaction can be targeted to decrease the metastatic potential of the tumor and provide improved prognosis. That's it. Thank you so much for listening. If you have any question, we'll take it.